Blood tests are widely used, but they are invasive, it takes a lot of time to get the results, and you cannot take blood continuously if you want to track how things are evolving. It's not like we are leaking out blood. But breath? You're already exhaling it all the time, and you can take as many exhalations as you need. It's not just more comfortable, you can capture information that would otherwise be impossible to obtain. Let me give you an example. Let's say you need to take chemo. It's a difficult situation. Chemo is very aggressive, so nailing the right dose can make a big difference. Your doctor tells you you need to breathe into a machine every minute while they are pumping chemo in your veins to check that you are metabolizing it correctly and you are getting the right dose. Could you do that? Would you want to make sure that you get the right dose? Now let's say that you have to take a blood analysis every minute. That would be very hard, and that's why it's not done. What you see here is a complete research workflow for breath analysis. You exhale, monitor the flow rate and CO2, and monitor exhale metabolites with a mass spectrometer. All instruments shown in this video are for research, none of them are a medical device. Non-invasive, fast, and capable of measuring thousands of molecules in just a few seconds, breath analysis opens possibilities for early screening, non-invasive diagnosis, drug monitoring, you name it. Police use breath tests to detect alcohol every day. Doctors, not so much. Why? In this video I will explain what's holding breath analysis back and how we are solving these problems. It's a long story, so there is an index in the description in case you want to jump to a specific section. Stay with me through the more technical sections. The conclusion might affect you sooner than you think. To turn breath into a diagnostic tool, we first need to identify biomarkers in breath. So, what is a biomarker? It's a molecule that we detect whose concentration changes with the physiological state or disease. Without good biomarkers, breath analysis only tells us the chemical composition of breath. Biomarkers tell us something meaningful about the body. The key is finding these special molecules. I like calling this biomarker mining. What kind of molecules make good biomarkers? The smaller and most common molecules tell us almost nothing. Water on Mars would indicate hope for life. That's a big deal, but here on Earth, it's everywhere. On the other extreme, your DNA is unique to you. In general, small molecules are common and vague, and big molecules are more specific. The conclusion is larger molecules are better candidates because they are more specific from a biological standpoint. Remember those days? That was weird. But we learned that lungs release very large molecules, even whole viruses. And that's great news for the biomarker mining community. Large molecules have a better chance of being biomarkers. Yet, clinical breath tests measure only very light gases like hydrogen, CO, CO2, or nitric oxide. That's strange. The lung can produce large and specific molecules, but only the small molecules are tested. Why? From a technical point of view, measuring the small molecules is easy because they are highly volatile gases. Detecting and quantifying large molecules in breath, that's a lot more difficult because their concentration is very low and they tend to condensate. Younger me thought, hey, that's an engineering problem. I'm an engineer, let's just solve it. This contradiction is indeed an opportunity. Some biomarkers have already been identified among the small molecules, so we should be able to identify a lot more if we can measure the larger molecules. That was more than 15 years ago. Older me, I underestimated how big this challenge was going to be. But let's dive into the basic principles. First, why are larger molecules so difficult to detect? There is a strong correlation between a molecule size and its volatility. Molecules in the liquid are constantly bumping and pulling into each other. To escape into the gas phase, a molecule in the surface of the liquid needs to be kicked hard enough to break free from the pull of its neighbors. Larger molecules are in contact with more neighboring molecules, so it's harder for them to escape. And it's easier to fall back. They recondensate, either forming aerosol or sticking to the walls of the instruments. In short, larger molecules are less volatile, they prefer to stay in the liquid, their concentration in breath tends to be very low, and they are lost due to condensation. If we want to target them, biomarker mining tools have to be designed for low volatility molecules. This diagram is a way to picture the relationship between concentration, volatility, and the effect of instrument performance on the type of molecules that we can detect. The vapor pressure is on the horizontal axis. It is a measure of the molecule's volatility. The vertical axis shows how much of the molecule is in breath. This line represents the phase transition. Above this line, we get aerosols or droplets, below it, just vapors. Exhaled water would be here, with a vapor pressure of about 59 millibars at body temperature, and very close to saturation. Ethanol, at the legal driving threshold, would be around here, 140 millibar of vapor pressure, and a concentration of 1 part per million. The infamous virus of an infected patient would be on the other extreme, with about only 6 viral copies per exhalation. To detect ethanol from a drunk driver, the limit of detection of a breathalyzer must be below this point. Detection limit is the minimum concentration of the substance required by the detector to produce a distinguishable signal, and this would be the required volatility limit. The volatility limit is the point at which molecules condense and are lost before they can even reach the detector. With a breathalyzer performance, we could detect molecules in here. But most exhaled metabolites sit somewhere over here. Imagine this was a biomarker, or this. To find it, we need to detect it reliably. That means improving the detection limit and the volatility limit. With better instruments, we will be able to interrogate more molecules and find more potential biomarkers. And today, that means mass spectrometry. Why? A mass spectrometer counts ions and measures their molecular weight with great precision. Some instruments are so sensitive they count ions one by one, and that gives us an extremely low detection limit. This is what mass spectrometry data looks like. In a spectrum, the horizontal axis is the molecular mass of the ions, and the vertical axis is the number of ions. Each peak shows the number of ions detected for that particular molecular mass. Zooming in, we can see that there are a lot of small peaks at the bottom. One of them might be the biomarker we are looking for. When we analyze breath in detail, it is a complex mixture with thousands of different molecules. That means thousands of peaks in the spectra that we need to quantify. And for this, we use high-resolution orbitrap mass spectrometry. All the technological approaches like ion mobility, gas chromatography, or the two combine, or even electronic noses, they are smaller, they are cheaper, and some are even portable, a lot more convenient, but without the resolution and the sensitivity, the data quality is just not enough for serious data mining, and they are extremely vulnerable to confounding factors. Data quality? Confounding factors? What is that? Why does it even matter? Let's illustrate this with an example of what biomarker identification looks like. We take two groups of people, one with the disease, one without, and measure the chemical composition of their breath. The result is a table like this. There is a row for each signal and a column for each person. And each value is the measured concentration of that molecule in that person's breath. For each molecule, we can plot the signal distribution for the disease group and the control group. If the two distributions overlap, that molecule can't tell us anything useful. If they are separated, that's a potential biomarker. How is this linked to the quality of the data? If your measurements vary randomly, these distributions get wider, they overlap, and we cannot differentiate them any longer. To recap, the tools to enable breath biomarker mining must produce high-quality data for large molecules which are low volatility and low concentration. The breath analysis pipeline includes capturing molecules in breath, ionizing them, delivering into the mass spectrometer, and analyzing the corresponding signals. And the quality of the data depends on the weakest link of all these steps. Where did we start? Well, you need to see the data if you want to improve its quality. So the natural first step was to develop a breath ionizer to ionize breath vapors and be able to detect them with a mass spectrometer. So we optimized secondary electrospray ionization, or SESI as we call it. Let me walk you through how it works and why it's ideal for this application. In SESI, a nanoelectrospray produces tiny droplets that evaporate and turn into a cloud of protonated water clusters. Breath is exposed to this cloud. Vapor molecules get protonated when they collide with a water cluster, and they are passed to the inlet of the mass spectrometer. In a way, SESI is similar to atmospheric pressure chemical ionization, or APCI. In APCI, a corona discharge produces high-energy ions which decay and form protonated water clusters that transfer their charge to the vapor molecules. But there is a key difference. High-energy ions, even if there are only a few, smash the molecules, producing fragments and exotic oxidation products. So each molecule leads to several peaks. If a spectrum is like a forest, the big trees in plasma-based and SESI spectra look very similar, one big protonated peak per molecule. But when you zoom at the bottom, where the hard-to-spot biomarker will be, the story is different. If you have several molecules and several peaks for each molecule, which peak is the original molecule? Which one is a fragment or an oxidation state? To what original molecule does each peak belong? There is no straight way to tell. With SESI, you still get simple protonated peaks at the bottom. Also, we can place the tip of the spray a lot closer to the MS inlet because we don't need to wait for high energy ions to decay. That gives us a very high concentration of protons and better ionization efficiency. This is a super SESI source. This is the nanoelectrospray, and here's the sample inlet, and this is the interface to the mass spectrometer. Super SESI is optimized for ionization efficiency and to minimize condensation losses, so we can detect larger molecules with low volatility. Nanoelectrospray is incredibly sensitive for large molecules and produces very clean spectra, but there is a price to pay. Controlling the nanospray is more difficult than just applying a voltage. That is why the rest of the Super SESI is dedicated to house all the controls. You could say it's an ion source and a robot in one. With this, we can take directly breath from the person and deliver the ionized molecules to the mass spectrometer in real time. So what do we see when we do that? The mass spectrometer produces several spectra per second. If we track the evolution of one of these peaks, we can see how the corresponding concentration goes up and down as we exhale. Signals go up during the exhalation and return to the background level when the exhalation is finished. But each molecule has its own story. Some start high and then drop. These are usually from molecules that are in the upper airways. Others start slow and rise slowly, likely coming from a deeper part of the lungs, just like CO2. And if you do multiple exhalations in a row, you'll see some signals climb or fall between breaths. The composition of breath is very dynamic. The study of how the composition of breath changes over time can be fascinating. You could study how aromas are released in the mouth and the nasal cavity and how they are perceived. This is relevant for the food and aroma industry. You could also study how inhaled substances behave in the lungs. For instance, we can track the washout profile of nicotine and other tobacco-related metabolites. But if our goal is to identify biomarkers, these physiological shifts add a lot of noise. They widen the signal distribution and blur the difference between healthy and diseased groups. We need to eliminate that source of variability. That's why we created Exhalation. Its mission is to guide exhalation maneuver and remove two big sources of variability. Let me show you how it works. Here, the subject is taking a breath test with Exhalation. Exhalation gives the subject a visual guide to match the flow rate and the exhaled volume. This means all exhalations are standardized. Exhalation also logs the time, CO2 flow rate, and exhale volume. Later, that data will be used to differentiate the lung fractions, align them, and compare the same lung fraction across all participants in the study. A lung fraction is defined with a CO2 and exhale volume thresholds. Using Exhalation data, we find the time for which the exhalation was in those thresholds. And then we use the same time tags to average the signal of a metabolite detected by the mass spectrometer. If we're interested in the upper airways metabolites, we can set the thresholds to 1% and 2% CO2 concentration. If we're interested in the deeper part of the lung, We can set those thresholds to 3% CO2 concentration and beyond. Those average values are the input to our data matrix. With exhalation, we get a lot of noise and variability, but that's just one bottle. There are other confounding variables. Let me explain what a confounding variable is with an example. You find a potential biomarker. Most patients have it. Controls don't. Sounds like you are on a good path until you realize it's a painkiller. That's a confounder. That's an obvious case, but there is another sneakier problem, and it has nothing to do with chemistry or biology. It's pure statistics. The better the instrument, the more peaks you detect. Sounds like progress, but here's the catch. The more peaks we measure, the higher the chance that some will look like a biomarker by pure chance. In a typical SESI orbitrap breadth spectrum, we might track about 8,000 peaks. If you set a significance threshold of 0.1%, probability tells us you should expect about 8 false biomarkers. And if you use combinations of signals, so-called fingerprinting, the statistical problems becomes huge. If 8,000 sounds like a large number, combinational numbers are a lot larger, way larger. They have factorials in them, and they explode rapidly even with very few signals. It becomes like a casino, but betting on all possible combinations. Here's the paradox. The better your instrument, the more likely you are to be fooled by randomness. Have you noticed there are several papers claiming to have identified biomarker sets that never replicate? When results can be replicated, it's often due to confounders that weren't addressed. You have to take confounding factors and statistical significance very seriously, otherwise you will end up hallucinating biomarkers in the datasets. That's why managing confounding variables carefully is an essential part of biomarker mining efforts. Good practices like randomizing batches, proper statistical analysis, multi-center studies, and so on are very important, of course, but unless we control variability and reduce confounders, those methods demand massive cohorts of patients, and huge cohorts aren't always available. After all, you only have so many patients in a hospital. Some scientists chop their data sets to fix this problem, keeping only the strongest signals. As an engineer working to make better instruments, I can understand it, but it hurts. You don't have to throw away data to keep the statistics clean. If you know what biomolecule is behind each signal, you can use your biology knowledge and the context of the experiment to eliminate confounders without losing sensitivity or statistical power. You can tag painkillers and other known confounders from the dataset. You can use controlled stimuli, a trigger event like taking a drug to see if the response is reproducible. That's a lot harder to be randomly reproduced. Also, if a group of related metabolites all sieve together in a way that makes biological sense, that can be more credible. The key to these strategies is knowing at least the molecular formula for each signal detected. That is why soft ionization isn't just about getting a pretty spectrum, it's about making sure we are incorporating molecular biology, not just doing blind statistics. If the ion source produces exotic fragments or oxidation products, the peak count explodes, randomness is your statistical significance, and the maze becomes unsolvable because you can't even assign molecular formulas. SESI, with no high energy ions at all, gives you a much better shot at finding biomarkers that are real, incorporating the knowledge of molecular biology. Okay, so that's how you get good data sets, but post-processing this still can be a challenge, even if we have good data quality. We're talking thousands of peaks, hundreds or thousands of samples, each with a particular time evolution. We need to assign the molecular formulas, and it's not as simple as one peak equals one molecule. There are isotopes and some adducts. That requires some serious computational power, and for this, the peak-by-peak software was developed. It synchronizes mass spectrometry and exhalation data, calibrates, groups isotopes and adducts, and assigns molecular formulas and signal intensities for the different Lamb fractions. automatically. And that's it. If we are biomarker miners, the tools that we need for breath collection and exhalation standardization, exhalation. For soft and efficient ionization of large vapors, super SESI. For mass analysis, Orbitrap. And for data management, peak by peak. And that's how we go from breath to high quality data that we can understand. That's it. Hold on. I've been talking about analyzing breath in real time, but there are groups doing some serious research with offline analysis. In offline breath analysis, you first collect the breath sample and then take it to the lab for analysis. Some researchers use bags to capture the breath, but the most common method is thermal desorption coupled with gas chromatography and mass spectrometry. Here's how it works. This is a thermal desorption tube. It has a trapping material inside. You blow inside the tube like that. This tube traps the volatile compounds from your breath. In the lab, the tube is heated to release those compounds and analyze them. It's convenient, and there are some fancy adapters to blow into the tube. You could say that the scientific community is split between offline and online teams. And the offline supporters have three main objections of online analysis. Objection number one, they say we cannot pre-concentrate. Their argument is simple. With a sorbent tube, you can blow into it multiple times to trap more molecules and improve sensitivity. This is what I have to say. We designed to avoid condensation because we know most molecules are lost to condensation. And the first thing you do here is condensate in the tube. Call it trapping, but it's condensing. From our experience, the lung is the best desorption ever made. Can you imagine trapping a virus in this tube and then heating it up and desorbing it in the gas phase? If you trap large molecules here, you'll never desorb them again just by heating. At best, you burn them. So our philosophy is simple. Don't trap. Don't condensate in the first place. Just let it go straight into the instrument. Objection number two, they say we don't have chromatographic separation. And this is a real issue. Without chromatographic separation, it is harder to identify which molecule corresponds to its signal. But chromatography takes a lot of time and requires sample preparation, and for those reasons it's better to avoid it. That's why we use high-resolution mass spectrometry and very soft ionization. It's the only way to get away with skipping the gas chromatography and keeping the identification power. Yes, it means using nanoelectrospray, which is more demanding, but in exchange, you see lung dynamics in real time, you get to understand and optimize process much faster, you cut the time from sample to insight from months to minutes, and more importantly, by removing all sample handling steps, you eliminate the confounding factors they introduce, not in the data analysis step, but in the very early phase of measuring. That is built-in simplicity for better data quality. Objection number three, they say bringing people into the lab to excel into the analyzer is not always possible. And they are totally right. And it's an important point impacting the statistical significance. That's why we develop our own offline breath analysis system. This design builds on prior work from other scientists who used bags with SESI. It's designed to minimize condensation losses. Breath samples are stored and collected in bags like this, inside stainless steel canisters like this. The canister has heaters, sensors, and valves inside to control the temperature and how we inflate when we exhale into the bag and empty the content of the bag when we put it into the analyzer. And the data produced with this is compatible with the data produced by blowing directly into the analyzer. This is important because the future is in combining online and offline analysis. Online real-time analysis is the gold standard. You can capture the full exhalation profile of each molecule, define the best sampling procedures, and provide rigorous quality control. Once those procedures are validated online, you can apply them to the offline system. That way you can reach far more people, scale up your studies, and achieve a higher statistical significance, while using online analysis as a quality control. These machines answer the question, what is the chemical composition of breath? But they don't answer questions like, what's happening to my patient? Or, is this drug being metabolized properly? So what's the next step? The main bottleneck is statistical significance. We simply don't have data sets large enough to beat the noise. That means we need to measure more people. There will be a tipping point. Once the database reaches a critical size to find some biomarkers and start offering clinical service, even if they are niche, breath analysis will be economically viable. New machines will pay for themselves, and the services they provide will generate more data, allow us to find more biomarkers for more clinical applications. It's a feedback loop. Bigger data sets lead to more biomarkers, more clinical service, more data collected. You get the point. Breath isn't just air, it's information. And with the right tools, we can read it. Breath analysis will happen. The question is, who will lead it and how soon? There's a race, and whoever builds a reliable breath biomarker database will define the field. Until that day comes, we will keep working in our workshops and labs. If your goal is to make breath analysis a reality, get in touch. There is a link in the description. If you find this useful, subscribe and like. That tells me to keep making more videos like this. See you next time.